G protein-coupled receptors, or GPCRs. Cell signaling pathways. Cell signaling pathways include the 4 steps we talked about previously, where the signal travels to the receptor, the signal alters the receptor, the signal is transduced through the cell, and the cell responds. We've already discussed steps 1 and 2, the different ways that the cell can travel to the receptor, and how the signal alters the receptor by its changing shape. Next, we will focus on number 3, signal transduction. Then we will add number 4, cell responses, to discuss complete cell signaling pathways. G protein-coupled receptors, or GPCR, are an example of how signals can be transduced across the cell membrane. At least 800 different genes, or 4% of the human genome, encode GPCRs. They are involved with sensory reception, behavior and mood, immune responses, homeostasis, the endocrine system, and tumor metastasis. Hyperactive GPCR can cause disease. 34% of all drugs approved by the Food and Drug Administration target GPCR. That is, the drugs bind to mutant GPCR and inhibit them, or bind to molecules in their transduction pathways and inhibit them. GPCR are also called serpentine receptors because they have 7 transmembrane alpha helices that snake back and forth across the membrane. Their outer domain has a ligand binding site, and their cytoplasmic domain has a G protein binding site. In the membrane, they are closely associated with G proteins and an enzyme, such as adenylyl cyclase. The illustration shows the rhodopsin GPCR, which is part of the rod cells in the retina, passing through the cell membrane 7 times. So if we start here at this NH3 plus end, we can pass through 1, 2, 3, 4, here's 5, 6 is behind, and then as well as 7, and then we come out this other end here, COO minus. As the ligand binds, as shown in image A, to the receptor changes shape, transducing the signal across the membrane. In image B, we see that makes the G protein binding site accessible. C. G proteins are peripheral membrane proteins. They bind GDP similar to ADP when inactive. When activated by a ligand-bound GPCR, GDP is replaced by GTP, similar to ATP. So you can see that now GTP can bind. The activated G protein diffuses in the interface of the membrane to an enzyme, such as adenylyl cyclase, phosphorylating and activating it. Once active, it converts ATP to cyclic AMP, CAMP, a second messenger, which leads to transduction within the cytoplasm. G protein-coupled receptors. The next slides show how transduction occurs with a GPCR. Initially, the GPCR is composed of a protein that passes through the cell membrane 7 times and has an inactive G protein docked to it. The G protein is composed of 3 subunits named alpha, beta, and gamma. Its alpha subunit is what binds GDP. So you can see in the image here, the GPCR is a protein that passes through the cell membrane. There's an inactive G protein docked to it, which we see here, alpha, beta, and gamma. And the alpha subunit is where the GDP binds. GPCR transduction step 1. So here is our inactive G protein. The first step is a ligand attaching to the GPCR, which changes its conformation, leading to GTP in red, replacing the GDP in the alpha subunit of the G protein. Step 2, once the GTP binds to the G protein, displacing the GDP, the alpha subunit of the G protein separates from the other two units, as seen here, triggering a cellular response. As we will see, that can involve secondary messengers. Step 3. The GTP is hydrolyzed to GDP with the release of a phosphate. You can see here now we have a GDP and a phosphate, and the ligand is released from the GPCR. Step 4, once the ligand is released and we get the GDP binding to the alpha subunit with the release of the phosphate, is that the alpha subunit rejoins the beta and gamma subunits, and the G protein becomes inactive in its initial state. So we can start with an inactive GPCR with a G protein. The ligand binds, which changes the shape or the conformation here, which displaces the GDP, allowing GTP to bind to the alpha subunit, which then separates from its gamma and beta, generating a cellular response. Once that happens, the GTP becomes GDP with a free phosphate. The ligand is released, and the G protein becomes inactive again. The preceding slides show how a ligand being taken up by a GPCR can result in the alpha subunit of a single G protein generating a cellular response. Generally, more than one G protein is activated, and the cellular response that follows from a single ligand includes a cascade that can lead to more than 10,000 enzymatic products. Some steps in the cascade involve adding a phosphate to a molecule, a process called phosphorylation. This changes the molecule's shape, which can then catalyze or inhibit a reaction. An enzyme which catalyzes phosphorylation is called a kinase. In the following slides, the reaction is catalyzed by a kinase. On the left, we have an illustration of a GPCR taking on a ligand and activating G proteins. So here's an inactive G protein, now active with a GTP, by the same process that we explained in the preceding slides. In this case, the GPCR activates 7 G proteins, as shown by these 7 blue arrows. I only drew 3 G proteins fully due to the space limitations, but assume we have these same activated G proteins all the way around. Each of these G proteins then travels to an adenylyl cyclase. So these are adenylyl cyclase. You can see each G protein traveling to one of them. It is bound to the cell membrane, and 3 of the 7 are shown traveling in the image. The G protein then binds to adenylyl cyclase, which you can see here. Here, 3 are activated by the binding of the G protein, and 1 is not yet activated. Adenylyl cyclase catalyzes the production of CAMP molecules from ATP. Since the catalyst, adenylyl cyclase in this case, isn't consumed by the reaction, large numbers of CAMP molecules can be produced at one site. Each blue arrow represents the production of one CAMP molecule, though the CAMP molecules in green are only shown on the rightmost activated adenylyl cyclase due to space. So you can see the production of the CAMP by these green molecules, and you can imagine them here as well. Each CAMP molecule activates a kinase. Kinases are enzymes which transfer phosphates through a process called phosphorylation. You can see the activation of the kinase in purple. Each kinase can activate 10 or more of the enzyme in the next stage. Catalysts are not consumed, so can catalyze many reactions. Only the amplification of one kinase is shown right here due to space limitation. However, the 10 shown kinases will activate 100 enzyme molecules. Finally, that amplification is repeated once again, activating 1,000 of the final enzyme molecules, which we're calling the enzymatic product. If there were 10 activated adenylyl molecules, this cascade would result in a 10,000 to 1 amplification of the signal from a single ligand. The enzymes in the final stage of this process catalyze a reaction in the cell. If the ligand here was epinephrine, the resulting enzymes would trigger the fight or flight response. In the liver, this would result in the breakdown of glycogen to release glucose for energy. Epinephrine is secreted by the adrenal gland, so is called adrenaline. At any stage in this process, from here on through this cascade, this process can be inhibited, which would end the process at that point. This drawing of the pathway was used in the first question of the 2022 AP exam. While students were not expected to know the details of the pathway since it was explained in the question, being familiar with it would have been a big help. Relate the steps shown here to what was shown on the previous slides, except for the chloride secretion at the last step. So we start here with an inactive G protein and a GPCR receptor that does not have a ligand at the binding site, and an inactive adenylyl cyclase. The ligand binds to the receptor, and what happens? The GDP is displaced and replaced by GTP, activating the G protein here. Once the G protein is active, remember we talked about how it can then travel to the adenylyl cyclase, which we see here. So that's what happens here, is that activated G protein travels to the adenylyl cyclase, and look, it's only that alpha subunit with the GTP. Here are the beta and gamma subunits here. Okay, so this activates, this binding of the active G protein to the adenylyl cyclase then activates it. Then what happens? That catalyzes the production of CAMP. That CAMP catalyzes or activates a protein kinase, which then leads to this chloride secretion by the cell, which we don't need to discuss. Okay, so this just shows one of the cycles of GPCR pathway that we covered in more detail in the preceding slides.